Good afternoon, good day, hello. I'm Lloyd Nackley. I'm hosting today's webinar, and I'll be talking about hydroponic plant nutrition. Briefly about myself, I'm an associate professor in the Department of Horticulture at Oregon State University, and my program focuses on nursery management and greenhouse production. And I'm stationed at the North Willamette Research and Extension Center, which is off campus near Portland, Oregon, if you're familiar with the area. And I'll queue up my slides in a moment. The presentation I'm giving is the second, and we have two more lined up already. For October 18th, the next webinar will be buckets. So Dutch buckets or Beto bucket systems, as well as the nutrient film technique, NFT. So we're going to go right into hydroponic systems after today's lecture, which is more an overall or theory about nutrition. And then the following week on October 25th, we'll be having deep water culture, which is a type of system, as well as ebb and flow systems. And Michael will be leading both of those lectures, those webinars. This project overall is sponsored by the Oregon Department of Agriculture through a specialty crop block grant, where we're entrusted and working on disseminating hydroponic education in Oregon. Today, I am focusing on hydroponic plant nutrition, and I'm kind of breaking up my talk into what I think is about 3 sections. 4 if you include question and answers. So first, environmental. I'm going to do a brief history, 10,000 years in 5 slides or something like that. And then I'll go a bit narrow into elemental. So looking at these essential plant nutrients, essential and beneficial. And then also some mildly technical, but some very specific look at how to supplement. So fertilizing, I'll talk about actually understanding the fertilizer label, mixing the tank, and then a few things beyond just the fertilizer solution that's important for nutrient management. On your screen, you should see a QR code. This is part of our feedback survey that we'd really appreciate you participating in. We do want to deliver content and information that you find relevant. We also want to know that you're learning through the process. So if you could take the time to scan it with your phone and pull that up, that would be great. We'll have that again at the end, but if you want to pull it up now and follow along, you're encouraged to do so. So thanks. So seeing no questions yet, or at least being interrupted by them, I'm going to start way back real briefly. So soil, I know this is a hydroponic, but I want to give context that soil really is our history. Soil and soil nutrition has been known for ancient agriculturalists for millennia, essentially. The idea of incorporating animal manures as well as green manures, so compost like legumes, has been written about, talked about, and studied really since the inception of agriculture. And what I focused on that slide also is that there's been a fundamental rebirth of this in the last 55 years. So I'll get to the 20th century in a second, but we see a disconnect for about 50 years where we really focused on chemicals. But our work at Oregon State University, through our many crop science programs, as well as our horticulture programs and small farms, focus a lot on cover cropping and reincorporating plant material into soils. When you look at the development of agricultural history, and I'm choosing Chinese agriculture in this example with the figure on my right, at least the purple graph, because there was a wonderful study about that. We see that in more modern times, so really the last 500 years, but a focus on the last 200 years, that fertilizers itself has become a big focus of how we're trying to increase our productivity in agriculture. And so that's what that figure shows on the left, is this huge time scale from 8,000 BCE to the beginning of the 20th century, and the different widths of the chart show the emphasis of advancements. Again, this was Chinese agriculture, but I found parallels definitely in Western agriculture as well. So the top one is fertilizer and the increase of technologies. Some of these that are these discoveries that are somewhat well known or very well known in the plant science world focused really between that 16th century and 18th century, this kind of reawakening or this renaissance of chemistry in at least Western science. And so I'm highlighting 3 of those studies because they really are fundamental to what we as plant scientists and agronomists think about when we're approaching plant nutrition. And so the first one on the top is Jean-Baptiste von Helmont and his study of a willow tree, really. And so that's the picture of the 2 trees. And there was this hypothesis. So this was around, I forget, somewhere in the late 1500s. The idea that the food or the nutrition that plants were eating soil, right? And so this concept really was, for thousands of years, that's what people thought. So if you put manure or compost into the soil, the plant was then taking it up and growing from the soil, which is partially true, of course, as we know today. But what von Helmont's study showed is that he weighed the plant in the beginning, and then he weighed it later, and lo and behold, the soil was the same. So the mass of the soil was the same. So there was something else that was actually feeding the structure of the plant. And at the time, he thought it was water. But 100 years later, or shortly later, people were focusing on the gas that was around the plant. And so there's a person named Joseph Priestley who did this experiment with mice or with rodents, where he put them in these little containers. And so this is the image on the lower left. And with a candle, the oxygen was consumed, or they didn't call it oxygen until Priestley, so that the air, the gases were consumed. And of course, the rodents suffered and suffocated. But if you put a plant in this chamber, then it cleaned the air, so they thought, and was regenerating this invisible source that was providing life. And so that became called oxygen after these types of experiments. And really just shortly later, a person named Jan Ingenhousz, who was acknowledged by a Google Doodle, which feels like our modern, like highlight pantheon. If you get a doodle, you must have been somebody. So this person, Jan Ingenhousz, realized or formulated that light was this fundamental aspect that was missing from Priestley's experiment. And so these 3 experiments are often thought to be kind of fundamental to photosynthesis, and then where we are today with understanding what plants need to grow. So if we think about soil as our history, farming in soil is what we're used to. And so it's a natural source of nutrients. If you take care of the soil, this concept of soil health or soil tilth is now a basic idea, a basic tenet that farmers think about, people farming in soil. If you support the soil, there are beneficial microbiomes, so bacteria and fungi that you can, in a sense, partner with to help grow your crops. Soil is a buffer. It's a huge insulator of temperature. So freeze and heat events are buffered and the roots are protected. And then we have this huge, broad, traditional-based knowledge. So Western, Eastern, Southern, and Northern people have been farming in soil for countless generations. And so we can build on that. And the last one is there's lower technical requirements. Yeah, this whole webinar is about hydroponics, and that's soilless-based production, right? So growing in water primarily. And why would we even consider that? Really, because if soils are history, then in our recent present, chemistry is what's been guiding the growth and the development of agriculture. And so I talked about those 3 historic discoveries. One of the most important discoveries of the 20th century was this process called the Haber-Bosch process. And this is the method or the process for pulling nitrogen out of the atmosphere under pressure and temperature, combining it with hydrogen to create ammonia. right? So the air that we're breathing is primarily nitrogen. It's 75%, but it's locked with this triple bond. So it's very, very stable. It's unreachable. And ancient agriculturalists and traditional agriculturalists today use legumes and the association with rhizobia and nitrogen fixing bacteria to pull this fertilizer, this most important element, out of the atmosphere. And it wasn't until the early 20th century, so 1910s, that these two German scientists figured out a method for synthetically doing this. And that's called the Haber-Bosch process, after their names, Fritz Haber and Karl Bosch. This discovery was one of the, or I say it was one of the most important because it enabled agriculture to expand rapidly. So it provided food for thousands, if not millions of people. It is also somewhat controversial because it's very energy intensive, and the excess of synthetic fertilizers has led to a lot of environmental degradation and runoff. But it's very important to know. So I think those that's where we're starting. That's, like I said, 10,000 years in a few slides. And so heading into hydroponics, which is this very supplemental based growing system, has a lot of strengths. So you can get higher yields. You can grow plants faster and larger in hydroponics. It's been demonstrated countless times that it is more water use efficient, up to 80 to 90% more water use efficient than soil-based farming. You are able to exclude chemical pesticides at a greater degree than soil-based farming. You can have a higher level of biosecurity, meaning restricting the crop from mammals, from avian pests, from other things that would impact food safety. You can have a higher control of losses because you can work on the timing of your crops. You can speed up or slow down your processes more than if you're fully dependent on the seasons. Non-arable land can be used, which is an incredible benefit. So urban spaces, brown fields, places with very poor soils for other reasons can now become cropping systems. And labor can potentially be more ergonomic, so higher up, less strength-based, opening up agriculture to many people who wouldn't have the physical attributes or characteristics to do traditional soil-based farming. Obviously, there are weaknesses. Expense is probably the number one compared to soil-based farming. It is expensive to set up hydroponics. Compared to planting seeds in a field. It is energy demanding. There's daily management tasks that are mandatory. And the one that I highlight at the bottom, because I think it's central to nutrient management, is that mistakes or accidents can cause catastrophic collapse of the system. So the idea is that if you're fertilizing in a field, you have this soil buffer, you can cause problems for sure, 100%. But there is no buffer, or there is much more limited buffer in the water. So any changes you make are experienced by the plant much more directly. Jumping right into the essential and beneficial nutrients. This is a concept, again, 20th century chemistry, quite a bit later than the Haber-Bosch process. So this concept of essentiality was coined and codified really by these two scientists, Samuel Arnon at Cal Berkeley, and then Lloyd Stout, Illinois. And I say that because there aren't too many Lloyds in the wild. So I definitely was surprised to see the first name, because I've seen Arnon and Stout, I don't know, for years, but I only found out recently for this presentation that was a fellow Lloyd. So these two folks, these two scientists, codified 3 criteria for determining whether an element is considered essential. And so I think, or I'm going to share these with you to give you hopefully a little leg up, a little trivia, but understanding why, really a background of why when we talk about the elements in a fertilizer bag, these are the ones selected. So there's 3 criteria. The first criteria is that the element needs to be necessary. So if you omitted the element, so if the element was not available to the plant essentially wouldn't grow. So it would either grow abnormally, or it would fail to complete a life cycle, so it might not flower, or it would lead to premature death. So it could grow for a brief time and then would collapse. And so that's the idea of necessity. So that's the number one criteria for essentiality. And I'll also say, since I am listing them in order, there is no hierarchy to these three. So if you see them in future lists, they can be mixed up. This is just the order that I chose to put them in. After necessity or necessary, it's irreplaceable. So the idea on this one is that you cannot substitute one element for another. So if this element is not present, a different one cannot fill the place of it in these plant processes. And the third one is direct involvement. And so that means that the element must participate or be involved in, is another way of phrasing that, essential biochemical processes, such as photosynthesis, respiration, enzyme activation, any of these processes that are essential for the plant to grow and develop. And if you have these three criteria for essentiality, then you come up with a list of 16 or 17 essential plants, elements in plants. And so there are more elements found inside of plants. If you did an analysis of the dry tissues of a plant, you would have more than these ones. But without these elements being present, the plant cannot fulfill those three criteria. It would not grow normally, it would not function, and you cannot substitute any of these in for another one. And so there they are at the bottom. And if you remember your, if you have a background in chemistry or high school chemistry, you'll see that it's carbon, reading from left to right, carbon, oxygen, hydrogen, nitrogen, potassium, which is a K in this case, calcium, magnesium, phosphorus, and sulfur. And they're grouped together by me as macronutrients. And they're called macronutrients because they're found in relatively higher concentration in the plant. And so what this bar graph is showing kind of a generalized amount of the percentage of these elements in a plant. So scientists take plant tissue, dry it down, grind it up, and put it in some sort of analysis system. When you look at the plant tissue, these ones on the left are found in higher concentrations, higher percentages. And then I've even put a box around the first three because they account for 90 to 95% of the dry weight. And what I think is kind of notable and interesting is that these three, carbon, oxygen, and hydrogen, are found or are gained or taken up just from the atmosphere. So CO2 from the air that we're breathing, and then hydrogen also can be taken up from the air or just from water, from the soil. The other macronutrients that would be found in the soil are this nitrogen, potassium, calcium, magnesium, phosphorus, and sulfur. And you see the numbers over those letters. They're much smaller than the first three, but still they're found in much greater concentration, times 10, like order of magnitude, or times 100, or times 1,000, to the other ones on the right. So the other ones on the right are called micronutrients, or that's what we think of them today. And so that would be chloride, Cl, Fe is iron, manganese is Mn, boron, zinc, copper, and molybdenum is the Mo. And so you see that, like, looking down at molybdenum, it is a very, 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 very small percentage of the plant. Yet, if this element wasn't available in the soil or nutrient solution, we would see that the plant would have abnormal growth and wouldn't be able to fulfill its growth and function. And so it's considered essential. There's this other group, and these are called beneficial elements. And so it's not controversial, but it feels like we're sort of on a precipice of discovery, right? We don't know everything. And we do know that these elements, So aluminum, silicon, cobalt, nickel, and vanadium also are very beneficial, very important to sustaining plant growth and vigorous growth. And occasionally, you or I, if you look into these, you'll see some of these. I've seen nickel and vanadium included in a list of essential elements. And so, so I've listed what these ones do. So aluminum is often used in at least nursery production, especially with hydrangeas to support flowering. Silicon is being used in a number of plant growth experiments for stalk strength, anti-drought, so drought stress abilities, antifungal, so reducing fungal pressure. Cobalt is essential. So this one's sometimes listed. Cobalt's essential for rhizobium. So if you're looking at legumes and nitrogen fixing bacteria, cobalt must be in the system. So that would be an indirect process, but still essential to plant growth. Nickel is helped to synthesize urea in grains, and vanadium, can be a partial substitute for molybdenum. But it's not just about the elements, right? So thinking of either soil or soilless solutions. It's not just having the elements present, it's the availability of the elements is the plant's availability or ability to uptake these elements. And that is highly dictated by the pH of the soil or solution. And so people might be familiar with pH and this chart with the wiggly geometric shapes down there. And so what that chart is showing is when those essential plant elements are most available. So at the widest point would suggest they're most available. And if you look up vertically, you should be able to see that range for most of the elements is in this 5 to 8.5 range, or right at 7.0 at neutral. So you're getting most availability at that range. And then when you see the tapering of the shapes, that would suggest that the element is less available at the pH. So pH is going to be strongly dictating what elements the plant can take up. And so the particular range depends with particular plants. So we speak in generalities, mainly because it's easier to understand and communicate. But when you end up growing a specific crop, you are going to be learning the specific range for that plant. A benefit of hydroponic systems is that you can tweak, you can change and adjust pH relatively easily compared to soil-based systems. At the same time, that means that the pH, and this is the buffering, can get out of whack much more quickly. So you have to watch pH a lot. I did list ericaceous, so people in Oregon at least are very familiar with rhododendrons, azaleas, and blueberries. So these plants that do like acid-loving soil. And I debated including that in here in this lecture since we're focusing on hydroponics. But because blueberries are one of the fastest growing crops in hydroponics, I thought it would be relevant. And so that comes to a surprise to some people, but it's because of this soil constraint for blueberries where they need this acid soil, and farmers work on how to manage that. But if you grow them in soilless solutions or a soilless system, you have a much stronger control of the pH and the availability. And so we're seeing large industry. So in California, Mexico, South America, and even a little bit in Oregon, heading towards soilless systems, so hydroponic systems for blueberry production. Deficiencies are when there's not enough. And so at first we were looking at the optimal range, and now we'll look at the suboptimal range. And so for the top 6 elements, so for these macronutrients, when pH gets low, when it gets acid, so that's why I've created that circle: nitrogen, phosphorus, potassium, sulfur, calcium, magnesium. So these macronutrients at acidic conditions, these nutrients are not available. Conversely, a lot of the micronutrients are unavailable at high pH. So just be aware of that, molybdenum being the exception there. The flip side would be toxicity. And so toxicity is when there is too much abundance of the nutrient in the solution. So this becomes a concern when elements interact with each other. And so if there is too much, in my example, manganese, you can lead to root death and leaf death. Aluminum, if you are using aluminum in the system, it can become toxic because it'll interfere with phosphorus and damage root cells. This idea of availability brings us to two other important discoveries in plant nutrition. The one on my left, which is the barrel staves, so the barrel that's leaking out with yields, is I think, a really common concept in farming and soil science, and this is the Sprengel-Liebig law of minimum. And I hyphenated it because in my research I realized that there was this strong collaborator, a researcher with the name Sprengel, whose name often gets left off of this law. So it's often just called Liebig's law of the minimum. If you've heard that one before, they're the same. The concept is that the growth of yield is limited by the availability of the scarcest nutrient. So it's that idea that overall growth is going to be limited by the one that is least available. And so it's kind of the same concept of a chain being as strong as its weakest link. And so in this one, the image is often used. This is like so commonly used, of yield being reduced because in this case, some element is at its lowest. And so that's where the yield goes down. This is a concept, I think, from the late 19th century, so 1800s, 1860s. More recently, almost 100 years later, when Stout and Arnhem were coming up with this idea of essentiality, a researcher named Mulder came up with a chart of interactions. And I think this concept is as important as understanding or being aware of the Sprengel-Liebig law of the minimum. Because with this Mulder's chart, which is this circle here with the arrows going one way and the other, it's illustrating the fact that in solution, these ions are being absorbed at different preferences and can interact either positively or negatively to impede the uptake of other elements. And so this chart, this Mulder's chart, shows antagonisms, which is where they're impacting negatively the uptake of another one in green. And you can see there are much, much more green than the stimulation where the element is actually positive for the uptake of another one. And so this idea of how nutrients interact is also critical for managing nutrient solutions. So I hope either of these is new to you or a good refresher, and there's a lot more information on both of them out there. And so when you get to this, so when you have this knowledge of management, you get to what I call the Spider-Man dilemma, which is this concept from the Marvel superhero. It's this classic line that before he dies, Spider-Man's uncle says to Peter Parker, "Remember that with great power comes great responsibility." And it's a concept of noblesse oblige, of that the more that we have in our control, the more we need to take care of what we're controlling. And so I think it's very important for people with experience in hydroponic systems or people going into hydroponic systems, which I think is often our audience, to be aware that once you move into these systems, you have to be paying a lot more attention. A concept that I've heard is like you're moving from a delivery truck to a Formula One car. Your plants are really dependent on you in hydroponic systems. And for that, I think it's really important to have a basic concept of the hydroponic solution. And that's, I guess, the nuts and bolts of this talk. So heading into the bolts part is a real basic concept. I had to create this slide and I thought about, do I need to or do I not? But it's a word that some people haven't heard before, and that's called fertigating. And it's a 100% made up word. based on irrigation and fertilizers. So if you hear me say fertigating, that means that the irrigation water has fertilizer in it. It is synonymous with nutrient solution, but I did want to put that concept there in case it comes up later or in your studies and you wonder what fertigating means. So here we go. There are a plethora of options for soluble-based fertilizers in the market. If you go to a hydroponic store, you will see racks and racks of different fertilizers and fertilizer solutions. If you go online, you will see even more. These fertilizers have a lot in common, and so I want to go through what their label shows to help you inform your decision of which to choose. The three things that I want to focus on the label are the guaranteed chemical analysis, is what it's called. That's on or should be on all fertilizer labels. The potential acidity isn't on all labels, but is an important characteristic of the fertilizer. And then, of course, usage rates. So the guaranteed chemical analysis, I think most gardeners will be familiar with this. At the top, there will be three large numbers that we all call NPK for nitrogen, phosphorus, and potassium, in that order. What's not commonly known, or at least thought of, and it's funny that we just keep passing it forward, is that it's not actually NPK, that these numbers are nitrogen, it is N, but the P is phosphate, the number is phosphate, so that's P2O5, and the K is potash, K2O. That's what these numbers are. And so it's broken down here, and it's not hidden on the label. It's just, it's not part of our lexicon or our vocabulary, but 20% nitrogen, 10% phosphate, 20% potash, and then other stuff. And if you ever wanted to calculate how much phosphorus or potassium are in the system, it is a very easy calculation. For phosphate, it's 10 times 0.043, or to get the phosphorus, and that's based on the atomic weight of phosphorus of the phosphate. And likewise for potash, it's 20 times 0.83. Just a little something. Also on the label, though, in addition to the macronutrients, are the micronutrients, right? So there's typically 6 micronutrients that get listed on a label: magnesium, boron, copper, iron, manganese, molybdenum, and zinc. These can be really important. You should make sure that if you are doing a nutrient solution, so if you are using any sort of these synthetic fertilizers to feed, that it should be a complete, or it should have micros in it. Most do, but every now and again, you might find one that doesn't. You can also find very specific fertilizers. That this one, which is relatively common, is called a CalMag or a CalMag Special. And that just means it has a higher proportion of calcium, cal, and magnesium. So this one would be 5% and 2, and that is quite a bit higher than many other fertilizers. Calcium and magnesium are critical, as many other or all the other elements for plant growth, but can be especially useful for hydroponic vegetables and fruits. So tomatoes, cucumbers, and some leafy greens, depending. Oops, I went 2. One thing to think about too is that not all nitrogen is the same. And so yes, the N is nitrogen, but it's broken up on the bag usually as in 2 forms, sometimes 3. And that this one shows 3. So that's ammoniacal nitrogen, nitrate nitrogen, and urea nitrogen. And sometimes urea and ammoniacal get lumped together because urea synthesizes to ammonia. But the big takeaway from the breakdown, and why there's a breakdown of the nitrogen forms, is that because as plants take up these different forms, there's a response to the water. So with the ammoniacal type, there's an exchange in the solution of a hydrogen ion. And so this will end up decreasing the pH. So as the root takes up the ammonium, it's releasing a hydrogen, which is pH. And so it's adding a hydrogen and decreasing the pH. On the other hand, if there's a high proportion of nitrate nitrogen, so the NO3, the plants will exchange a hydroxide, so the OH, and this will go out into the water solution, and that will increase the pH over time. In Oregon, where it's often cool and wet, you can have ammonium toxicity. So it can be a problem if there's excess ammonium in the system, because the plants cannot process it, they cannot store it. So that's something to be aware of when you're looking at it. Many fertilizers will have this on their bag, and so this is the calcium carbonate equivalents, or basically how acidic, what's the acid forming potential of the fertilizer. And so it's something to look at. It's not on every bag, I'll say that, but it's something to be aware of. In a soil-based system, this becomes something you have to think about for sure ahead of time, because it will dictate how much lime or liming type agents you need to put in the soil, or it will affect which fertilizer you're going to use. You can switch to an acid forming or a base forming fertilizer. Again, in a nutrient solution, it is a lot easier to make quick adjustments. And so really common products are things like pH up and pH down. And these are acids or bases that can be mixed into the solution to get the appropriate pH. The last thing that's critical on a label, that is critical on the label, is the usage rate. And so you'll find this one's a bit blurry because I tried to zoom it in so you could read it. It's actually from a fertilizer bag. But these are the parts per million N. So they're based on nitrogen, the ppm N, for your crop at a certain concentration. And so it usually, or can be, given in an EC value, so an electrical conductivity value, as a way to check it. Also, most of the bags will have injector settings, so to let you know how many grams you need to add if you're injecting at these different ratios. So the examples are 1 to 15, 1 to 100, 1 to 200. And that's like how concentrated your stock tank is going to be. It's a wide range, depending on the crop. Veggies are typically lower. Common floriculture crops a little higher, so 200 range. And then hemp or cannabis grown stuff can be higher. Same with fruiting crops. A few takeaways, and I don't know if this is too nitty-gritty, but I think it's important. Again, so picturing back to that Mulder's chart, the one with the antagonisms and the synergies. Phosphorus, so balancing your P. Phosphorus, we're learning recently. I remember I said that we're still discovering things. Phosphorus, we think now, last 10 years, maybe 15, 20, really recently, that we're over-fertilizing with phosphorus. So most plants can do a 2-to-1 ratio. So that's what this P, remember, so 20 to 10, so a 2-to-1 ratio is usually more than enough. But there's research happening currently looking at 3-to-1, so really lowering the phosphorus, or even lowering it altogether. And that's what these bar charts are for, is showing the results of phosphorus at different times. So nitrogen at 150, phosphorus down at 50, so 1 to 3, versus almost a 1-to-1 in this case. So I just use those charts to show how scientists are currently looking at plant development over time and how much phosphorus needs to be applied. So lower phosphorus than is recommended is the current recommendation. Similarly, potassium, we're learning a lot about how to balance potassium. A lot of crops will develop fine on an equally balanced potassium to nitrogen, so like a 20-20, right? So this is a 20-10-20 So equal, that would be a 1-to-1 ratio. But there are exceptions for sure. So we know azaleas like a 3-to-1, so much higher nitrogen. Begonias 2-to-1. Leafy plants 1.5-to-1. And then on the flip side, we have cannabis, which is a common greenhouse grown crop in Oregon, which is the opposite. So higher potassium to nitrogen ratios, at least at certain times. Potassium also, because it's a monovalent ion, so that's this K plus, can be preferentially uptake. So there can be competition with other ions, divalent. So calcium is a 2+, magnesium is a 2+. So magnesium deficiency can occur if there's too much potassium, and that has to do with all ion uptake in the roots. So again, Mulder's chart, that concept that in solution, there's all these essential elements, but the preference of the plant depends in part, in this case, on the size of the element and the ease of uptaking that ion. So if you start seeing magnesium deficiency symptoms, it could be a result of potassium and not that there actually isn't enough magnesium in the system. And last one, and I will click this fast, so I apologize, but just the idea that the timing of fertilizer matters. And so there are single step fertilizers or single bag, single step fertilizers where you'll just mix one bag, throw it in, and that's what you'll use for the whole development of your plant. There's also multi-step. So this company, General Hydroponics, has different stages where they're looking at how, in this case, nitrogen is being reduced at bloom, reducing nitrogen later on. So thinking like tomatoes can impact the fruit quality and yield, with lower nitrogen later on being better. So you're basically kind of forcing or suggesting the plant should decrease its leafy production and increase its fruiting production. And then this is just a 3-step product, but there are many fertilizers out there that are multi-steps, like 6 and more steps. So really targeting the fertilizer level or the element level at different stages of the plant. Just a few minutes left, but I want to kind of go over the how-to, right? So basic, like I was saying, the very basic is you choose a fertilizer, you mix it in your tanks, you inject it, and you monitor. And this is an actual fertilizer setup at a commercial grown greenhouse. They buy the fertilizer in on the pallets, they mix it in a couple different tanks, and they inject it, and then they go. You can make your own nutrient solution from scratch, and there's a lot of resources online and in textbooks on how to. The basic is called a modified Hoagland solution, and that's named after a professor, I think maybe at UC Davis, whose name was Hoagland. Sometimes it's called Hoagland and Arnon solution, and that's looking at taking these different individual constituent salts and mixing them together. If you do this, you need to have multiple tanks because the solution or the elements will precipitate at high concentration, meaning they'll form in a tank and settle out and become unavailable to the plant. This is a public resource that's called eGrow. There are a huge amount of uploads to this website and recipes and recommendations on how, if you want to. I'll say this right now. If you're just getting into hydroponics, then I do not recommend you start with the Hoagland, like mixing your own. I recommend starting with some pre-made solution and then working your way backwards. It's a little safer. The mixing order is important. So if and when you're mixing, don't do this on the top. So you'll choose your target ppm, your target parts per million. The idea is fill your tank part way up, so a third to half. This can be eyeballed, it does not have to be precise. Then add your solution, then add your water, and make sure that you test it with an EC pen or electrical conductivity pen before you put it out on your crop. That's critical because it's easy to make mistakes, and mistakes can have catastrophic effects. But the way to mix is this way. So you add some water, stir it in, and add some more water. The reason that that's so important is because continuous use, so as the water evaporates, the concentration will increase. Conversely, as the plants uptake the solution, the concentration will decrease, right? And so a good rule of thumb, oh, it's actually on my next slide, but this one. A good rule of thumb is that if you are running your solution for basically more than a week, you may need to replace and you will likely need to filter, because nutrients are being pulled from your system and the roots are releasing compounds that we just call them exudes, root exudes, into the solution, which can decrease growth as well. So about a week in a big production system of continuous running. This would be an NFT that Michael will talk about in coming weeks. How big should your tank be? It should be large enough to run a whole job at once. It should be opaque. So buy a tank that is dark to decrease algae growth. Something that is covered. So we talked about, I mentioned biosecurity in the beginning. We don't want something crawling on the top and dropping in and dying, or just crapping in your water. So those things, or swimming in. So covered, dark, and large, and then circulating. So keep something that mixes. Agitated in your tank is best. Where? Okay, good. The hose, the suspension hose, should not be on the bottom because fertilizers can precipitate and settle out. And you should inspect it regularly. So once a week. Like this is your lifeblood for your system. This should be a critical part of your production and not something that just gets left and not paid attention to. Monitoring can be complex, like these systems here, where a computer is measuring and dosing. So measuring, responding, dosing automatically. That is one option. I think we might be talking to people on an entry level, and monitoring still can be done very well, very effectively, and not that expensively, meaning a couple $100 with handheld pens. So this would be a pH and EC meter, and you would measure the solution pH and EC regularly, so daily at least, or in line, so where you would put the probes in your tank. Again, a few $100, and you can be monitoring pH and EC. 3 last things to cover that do not have to do with the solution, but are important to the nutrient solution. So I added them. 1, I said that carbon is one of the largest constitutes, most essential nutrients or elements to a plant. It is accessed through the air, through carbon dioxide and photosynthesis. The atmosphere has around, it keeps going up as we know, but around 400 parts per million. Plants optimize around 700 to 800 or more parts per million CO2 because the enzyme Rubisco evolved in a time when the atmosphere had much, much higher CO2 levels. So enriching with CO2 generally leads to increased photosynthesis, increased growth rates, so you have earlier maturity, maybe quicker crop turns, increased flower production, and increased water use efficiency. So if you are enriching with carbon dioxide, your plants may be able to they should be able to uptake their nutrient solution more effectively because they can grow and metabolize more quickly. That's done in a couple of ways. 2 that I'm highlighting are compressed CO2 tanks. So much like if you have a gas grill, you can get tanks of CO2, just like you have a tank of gas for your gas grill at home. You can get tanks of CO2 and burned and release it into the atmosphere, or actually just release it, just a little gas vent. The other one that's cheaper is if you get some sort of hydrocarbon. So you are then burning some sort of gas, and it is releasing CO2, and that can create impurities, but it's a lot cheaper. So if you have some tank, and these are CO2 burners, so they're burning a gas and CO2 is being released. CO2 is really important in your greenhouse to nutrient uptake. Another one that's critical is the temperature of your tank. It's baby bear here. Too cold on a hot day can cause leaf wilt. So if you have your tank underground or in the shade and your greenhouse is getting hot and your tank's 50 and your greenhouse is 70, that can cause stress for your plant. Flip it. Too hot of water, if your tank is in the sun and it's absorbing temperature and your greenhouse is 70, your tank's 80, will also be a problem. And a problem is in addition to this, and it's the next slide, is the oxygen load can go down when the water gets too warm. So here's an example from a tomato, big boy tomato, grown at different tank temperatures and the different absorption of the nutrients. And we see that potassium at the top. Potassium here is more affected by temperature than some of the other elements. They are all affected. You can see a peak right around 77 degrees. So this is in Celsius, but I converted it. So around 77 degrees for tank temperature for macronutrients. And similarly for micronutrients, we're seeing the effect of tank temperature. The last one that I'm going to cover before I take questions is oxygen. So the dissolved oxygen, which is regularly known as DO, in the water has a major effect on root activity and root function, and therefore nutrient uptake. So this is indirect, but it is so important in hydroponics that the solution has sufficient oxygen. The recommended range is around 5 to 10 parts per million. I have seen recommendations much higher, but the consistent across the different crops is around that range. The way to enrich your tank is pretty easy. You can do it with a water return actually spilling into your tank. So think of like a little fountain. So on the return in your tank, it spills in, it breaks the surface, and it adds oxygen. In addition, air stones, like you might see in a fish tank, or if you're more familiar with fish tanks, air stones are used. There are definitely systems that are promoted called nanobubble systems, where they're trying to create nano, so very small bubbles throughout the tank, and that's done through pumps and gas burners. Similarly, we found that just adjusting the pump that's in the tank to circulate can be used to create water and turbulence that will enrich the system with oxygen. So dissolved oxygen is critical, as is CO2 in the greenhouse or the atmosphere, and the temperature. Those will all affect your nutrient uptake. Winding down or winding back, I think my biggest takeaway, hopefully, is for you to be a good grower is to have some sort of feedback loop. So you'll make a decision, you'll select your fertilizer, and that can be based on the literature, based on this talk, based on what you find in the research. The next step that's critical is monitoring. So buy yourself some EC or pH pens, get an inline system that monitors constantly, have something that logs to your phone or your computer so that you can see how these critical values are changing throughout your day. Watch your plants. Plants are the phytometers, as sometimes they're called. They're the fundamental thing that's telling you how the conditions are. So we have, and I could have a whole talk just on nutrient deficiency diagnosis. It wouldn't fit in this one talk, but looking at those spots, the yellowing, where's the yellowing, the browning, sending leaves in, sending water in to labs for analysis. So that's that critical monitoring phase, and then the response. So then making decisions based on the information that you have, going back to the top for selection. When we're in these hydroponic systems, the plants are in our hands. They're like goldfish in a bowl. They are without their basic soil. And so the responsibility is on us. And there are resources out there for you. And that's all that I have today. And I have a few minutes, very few, but I'm happy to take questions if anybody has them. Again, we have this QR code here. We truly would love your feedback. And thanks for being here.
Thanks, Lloyd, for a great webinar. I am wondering, because of the great power that comes in managing nutrient solutions in a hydro system, are there particular crops which are more robust to fertigation mistakes than others?
That's a great question, Gail. Not really, which is funny. There are different media, so not all the systems use just water. So in a lot of hydroponic systems, growers are using coir, so the coconut fiber. Some are using perlite, some are using rock wool. And over time, these substrates can create some buffering, especially the organic ones like coir or bark, which is uncommon, but in nursery systems, it's more common. So you can get a little bit of buffering. Likewise, the larger plants do have more resilience. So not necessarily the crop, but the crop stage. So like tomatoes would be very vulnerable early, but if they're a big crop on a vine, they have some stored resources that might make them more resilient to changes. And again, it will also depend on the change too. So if you are creating deficiency systems because the pH starts going down, the plants will yellow. They won't necessarily die. But if you put, way too much fertilizer, some sort of toxic levels, that can create necrosis or death much more quickly.
Thanks, Lloyd. I have one more question. Folks that are online, please feel free to ask your questions as well in the Q&A pod. If you make a mistake and there is excessive nutrient release in the fertigation system, what do you do then? And if this requires a longer answer, then you can, in just like a minute or two, then feel free to refer me to a resource.
Sure. No, I mean, if you make the question, so if you make a mistake, what's the response? I think it will depend on how quickly you catch it. So if you see that you've over fertilized, you measure it before it goes out, you can dilute, right? So you'd have to remove some of your stock tank and dilute down. And that's really common, right? If you somehow catch it from noticing that your crop is being decreased, you will want to switch out your tank solution. And I guess the larger question is where do you, or I think a larger question is, where do you dump your solution? So if you have this concentrated fertilizer solution, 200 times what a plant would need, so that's like a 200 to 1, you shouldn't just dump that right in the gravel. That's going to go into the ground and into groundwater and run out. So pairing your system, depending on how large this is, like if we're talking a homeowners and it's maybe, a 50-gallon tank, you could probably distribute that on your lawn or something with extra irrigation water. But if we're talking commercial levels, there need to be some sort of catchment tanks, biofilters, wetlands that are used to treat this extra nutrient load before it goes off-site, because otherwise we're contributing to environmental problems.
Thanks, Lloyd. And there's a question in the chat. Matthias wondering, what is the most common mistake you see growers who are starting out make?
The most common mistake, I think, is, let's see, most common. It's probably waiting, probably not doing that feedback loop. So kind of waiting for the situation to fix itself. And again, I'm being specific to hydroponics. I think that maybe we get used to growing in soil systems where there are natural fluctuations of the microbiome. But if you start seeing a declining growth, the detrimental changes can happen exponentially. So changes can happen really fast. And so checking daily and multiple times a day if you're a commercial grower is what I recommend. So just being observant and being willing to make a change.
Well, thank you, Lloyd, and thanks all of you out there for attending today. I just wanted to remind you that we have 2 more hydroponics webinars coming up in the 2 upcoming Wednesdays, October 18th on bottle buckets and NFT, and then October 25th on deep water culture and ebb and flow. And if you want to register for those webinars, they're posted on our lab website, gardenecology.oregonstate.edu. I also dropped Lloyd's website in the chat as well. And thank you everyone so much.
Thanks, everybody.